Hello friends, I Dr Minal Harde from Department of Pharmaceutical Chemistry, PE Societies, Modern College of Pharmacy, Nigdi, Pune. Welcome you all. Today we are going to see in detail on topic column chromatography. Column chromatography is a technique employed for separation of components of mixture by continuous distribution of component between two phases, either it is mobile phase or stationary phase. When chromatography is carried out in a column, it is called as column chromatography, or otherwise it is also known as gravity chromatography. Column chromatography was developed by the American scientist D.T. Day in 1900. Then after that, Russian botanist M.S. Sweat in 1906 used adsorption column in this investigation for separation of plant pigments. There are various types of column chromatography such as adsorption chromatography, partition chromatography, ion exchange chromatography, and gel chromatography. Now, what is the principle of column chromatography? When a mixture of component dissolved in a mobile phase is introduced into the column, the individual component move with different rate depending on their relative affinities. The component with lesser affinity towards the stationary phase moves faster and is eluted out of the column first. The one with greater affinity towards stationary phase moves slower down the column and hence it is eluted later. Thus, the components are separated. Some common terminologies in the column chromatography, such as stationary phase. What is stationary phase? It is also called as adsorption, adsorbent. It is a solid. Then mobile phase. It is also known as solvent or eluent. It is a liquid. Sample. It is also known as adsorbate, which get adsorbed on the stationary phase. Then elution. Process for removing the component from the column is a process of elution. Eluate means the separated compound which is obtained from the column. So these are some common terminologies. Now we will see how the operation happens in the column chromatography. In column chromatography technique, the adsorbent packed in the glass column and mobile phase that moves slowly down through the packed column. A solvent used as a mobile phase is called eluent. A component attracted more strongly by the mobile phase will move rapidly through the column and elute from or come from the column dissolved in the eluate. In contrast, the component more strongly attracted to the stationary phase will move slowly through the column. Then experimental aspects of column chromatography. So here first we are starting one by one the components of the column chromatography. So first is the column. In that column, chromatographic column obtained commercially with the spring-loaded stopcock as shown in the diagram with a sintered glass disc at the bottom, which prevents the elution of stationary phase from the column. Then length of the column is most preferable is in the ratio of 5:1 or 8:1. And for the more efficient separation, we require the more length of column as compared to diameter. Generally 100:1 is preferred. Then column material is made up of good quality neutral glass, since it should not affect by solvent, acid or alkalis. A stopcock is fitted on the column is desirable as it allows control of flow of solvent. A small plug of glass wool or a porous plate is also present at the bottom, which support the adsorbent or stationary phase. This is the picture of the glass column. Then we will see detail about adsorbents. So many solid inorganic and organic substances are used as adsorbents in column chromatography, such as silica, alumina, calcium carbonate, calcium phosphate, magnesia, starch, etc. Alumina is generally suitable for chromatography of less polar compounds. Silica gel gives good result with the compounds containing polar functional groups. Adsorbents are classified according to their activity, such as weak adsorbent, medium absorbent, or strong absorbent. Weak adsorbent include sucrose, starch, inulin, talc, etc. Then medium adsorbent include calcium carbonate, calcium phosphate, magnesium, magnesium oxide, calcium hydroxide. Or some strong adsorbent include activated silica gel, activated alumina, activated charcoal, activated magnesia, etc. Then what can be the ideal properties of adsorbent? Here particles should have uniform and spherical shape. It should have high mechanical stability. It should be inert, insoluble in the mobile phase, colorless. It should allow free flow of mobile phase. Then it should freely available and inexpensive. It should be useful for separation of wide variety of compounds. So this should be the ideal properties of the adsorbent. Then how to select the stationary phase? So selection of the stationary phase is very important, as success of chromatography depend upon proper selection of the stationary phase. So the selection depends on some factors, such as removal of impurity should be there from the stationary phase. Then number of component has to be separate on the stationary phase. Then length of column should be greater as compared to diameter. Then affinity difference between components should be there, and quantity of adsorbent should be proper. So all are the selection of the stationary phase. Then how to prepare adsorbent or stationary phase? So adsorbent require activation before use, because moisture get absorbed on the adsorbent and it can interfere in the separation. That's why this can be achieved by heating, where adsorbent loses water and other adsorbed material. Generally there is an optimum temperature for activation. For silica gel we require 100 degrees Celsius for 1 hour, but for alumina we require 400 degrees Celsius. If we are keeping long time heating to the stationary phase, then lose of activity can be possible. Next is the preparation of column. How to prepare the column? So generally we have to take the glass tube with the bottom of the column should have center glass, then packed with the glass wool or other adsorbents also can be possible. Then we can use the developers. There are the components or reagent which are used for the production of color for colorless substances. Example: hydrogen sulfate, ammonium sulfide, potassium ferricyanide, potassium thiocyanate, etc. Solvent used generally. This solvent used in the color chromatography helps to transfer the mixture into the column. Then separation of zones to the highest possible means separation of all component at its highest level is also possible, and complete elution through solvent is also possible. So mostly we are using the solvents such as according to their polarity. So most non-polar is the cyclohexane, after that carbon disulfide, ether, benzene, toluene, ester, alcohol, chloroform, acetone, water, pyridine or organic acid. So based on their polarity we can use the solvent, depend on our sample nature. Then how to pack the column? So packing of column is of two types: dry packing, it is also called as dry filling. Other is the wet packing, it is also called as wet filling. So starting with the dry packing, in this the required quantity of adsorbent is poured as fine dry powder in the column, and the solvent is allowed to flow through the column till equilibrium is reached. Trapped air is removed by tapping. The demerits of the dry packing is air bubble are entrapped between mobile phase and the stationary phase, and the column may not be uniformly packed. Or the another demerit is cracks appear in the adsorbent present in the column. Hence flow characteristics and clear band of separated component may not be obtained. Another is the wet packing technique. This is the ideal technique. The slurry of adsorbent with the mobile phase is prepared and is poured into the column. The stationary phase settles uniformly in the column and there is no entrapment of air bubbles. There will not be any crack in the column of adsorbent. The bands eluted from the column will be uniformly and ideal for the separation. This is the picture which shows the wet packing of the column in the column. Then application of sample, how to apply the sample in a column which is already prepared. So application mostly done from the top using a small pipette whose tip is placed against a wall just above the surface of adsorbent. Another method is use of one or more small discs of volatile solvents, soaked filter, or we can use a soaked filter paper or place at the top of the column. So such techniques we can use for application of sample. Then detection, how to detect the separated sample. The detection of colored component can be done visually. The different color bands are seen moving down the column which can be collected according to its fraction. But for colorless component, the technique depends on properties of components and monitoring the fraction by TLC and UV light. Then illusion of sample. Here in the picture we are seeing that A, B and C. These are the three components. So one by one, according to their color intensity and color, we are separating the fractions. So illusion of sample is possible like that way. Then analysis of sample. So whatever the pure component we have collected, we can analyze whether it has mixture or it is pure. So we can check it by applying on the TLC plate. Then thank you. Here we have finished the part 1, and in the part 2 we are going to see a detail about. Thank you so much.